In today's lecture, we will talk about applications of two-dimensional electrophoresis. If you recall, in the previous lecture, we started discussing about some case studies about two-dimensional electrophoresis applications. So, as you have studied in this module, what are different type of workflow involved in performing two-dimensional electrophoresis experiments. We have gone through step by step, starting from the protein extraction, doing the quantification, isoelectric focusing, which separates protein in their first dimension, followed by doing the equilibration, preparing the strips for second dimension separation, and then you separate the proteins on the SDS page based on the molecular weight, followed by stain the gels to see the visualize the spot, and then scan those images, analyze that by using various software, and then perform some statistical analysis to obtain some biological insight. Now, this workflow remains same regardless of whatever application one want to use in their different type of biological questions. I started discussing about two-dimensional electrophoresis application in the last class. I gave you an overview of the 2D at that time. So, let us continue our today's lecture from the same theme, and let us discuss some more case studies on how people have employed two-dimensional electrophoresis, the power of this technique to resolve thousands of proteins and compare those for various differential proteomic applications. So, in today's lecture outline, first we will continue our case studies on conventional two-dimensional electrophoresis. We will talk about a study based on the drug treatment on malaria parasite Plasmodium falciparum. We will then take a study on the plasma proteome analysis of SARS virus. So, let us start with the first case study towards a proteomic definition of coartem action in Plasmodium falciparum malaria, a study by Makanga et al. In 2005. So, as you know, each year hundreds of millions of new malaria infection cases result in over 1 million deaths worldwide. But due to the lack of effective vaccine and widespread resistance to the anti-malarial drugs, still lot of deaths are happening, and the malaria problem is still posing challenges for its control. The anti-malarial therapy of chloroquine and pyrimethamine, these have not been able to control the mortality rate because of the anti-malarial drug resistance development. So, therefore, there is urgent need for identifying new drug targets as well as understanding the course of the action of these drugs by applying various type of high throughput techniques. So, in this paper, authors have discussed how two different drugs which are effective for the antimalarials can be studied for looking at the proteome changes in the Plasmodium falciparum parasite. So, coartem is a combination of artemisinin derived artemether with lumefantrine. How these two drugs behave and how the proteome changes occur due to the action of these two drugs were studied in this paper. So, authors applied proteomic approaches, the two-dimensional electrophoresis, to study the proteomic alteration of each of these drugs. So, these drugs are applied as a drug of choice for all cases of non-severe malaria worldwide. The artemisinin drug action is mediated specifically through its endoperoxidase moiety. However, the more detailed mechanism of action of these drugs are still unknown. So, the purpose of this study was to investigate the action of two active components of new antimalarial coartem, artemether and lumefantrine, on human malaria parasite Plasmodium falciparum. And authors tried to look for the alterations in parasite proteome which were induced by each of these drugs. To obtain the insight of the proteomic alteration, they separated the proteins on the two-dimensional electrophoresis gels and compared the response of these proteomic alteration based on these two drugs, and then they identified certain proteins which were either commonly expressed due to these drugs or they were differentially expressed due to these drugs. So, certain proteins were found to be commonly upregulated due to both of these drugs, and certain proteins have the different patterns. But before looking at the proteomic alterations, authors first determined IC 10, 20, 50 and IC 90 values for both the drugs ARM and LUM. We will use the abbreviations now for artemisinin and lumefantrine, and effect of these concentration of drugs on parasite growth over 24 hours was characterized. As you can see the growth curve in the slide. So, synchronized ring stage parasite cultures were harvested over 24 hours period after the exposure to the ARM and LUM. The parasite growth was determined by using hypoxanthine uptake assay. After establishing the culture conditions and the drug concentration, then authors looked for the proteomic alterations. So, first of all, they did the fractionation of the P. falciparum proteome, synchronized parasites which were isolated from the host erythrocytes, washed those initially and then solubilized that in a Tris buffer recipe. The Tris insoluble fraction was further subjected to extraction in the urea based lysis buffer. Once protein extraction was done, then authors used IPG strip of pH 3 to 10 range for the first dimension separation of protein in the linear IPG strips. After the IF was done, they equilibrated these IPG strips and then applied that on 12.5% of the vertical SDS gel. After the second dimension separation based on the molecular weight, then these gels were stained with the silver or Kumasi brilliant blue stains. So, by employing two dimensional electrophoresis and comparing the gel images by using the PDQuest software, authors were able to see that there is a differential proteomic response which is drug specific. So, quantitative analysis of the altered protein expression levels following exposure to the ARM and LUM were analyzed, and then those protein spots which were differentially expressed and statistically significant were further subjected to the mass spectrometry based analysis. So, the comparative analysis of 2D gels from untreated and drug treated parasite protein fractions provided direct and distinct alterations in parasite proteome following altimether or lumefantrine drugs. Certain proteins were identified, few of those showed common response due to both drugs. However, there are certain proteins which showed opposite trends due to each of these drugs. Proteins such as membrane associated calcium binding protein was upregulated in both the drugs. Aspartic proteinase was also upregulated in both the cases. Heat shock proteins such as HSP 60, 70 and 90, those were upregulated due to both the drug treatments. There are certain proteins such as enolase, fructose bisphosphate aldolase and phosphoglycerate kinase. These proteins were down regulated in artemether treatment and up regulated in the lumefantrine treatment. So, interestingly, the ARM treatment resulted in the more than threefold down regulation of the glycolytic enzymes such as enolase, phosphoglycerate kinase, fructose bisphosphate aldolase and glyceraldehyde 3-phosphate dehydrogenase. The expression of the same enzymes were also upregulated more than threefold due to the lumefantrine treatment. However, there are certain proteins such as the stress responsive proteins like heat shock proteins, which were commonly induced due to either of these drug treatments, which looks like a general stress response as compared to any unique response to the given drugs. So, from this study, the major findings were that authors successfully investigated alterations of parasite proteome induced by two component of coartem, artemether and lumefantrine. By using proteomic approach, they investigated specific and non-specific effects of two anti-malarial drugs in pharmacological relevant conditions. Expression of certain proteins were quite interesting, including a membrane bound calcium binding protein, which was upregulated due to artemether and lumefantrine treatment. The study also established a relationship between the pharmacologically relevant concentration and time of exposure for the two components of coartem. Let us now move on to case study 2, plasma proteome analysis of severe acute respiratory syndrome SARS, a study by Chen et al. In 2004. So, the purpose of the study was to perform a comprehensive plasma proteome analysis of severe acute respiratory syndrome and its comparison with the healthy individuals. In this study, authors employed conventional two-dimensional electrophoresis, analyzed those images from the treatment with the controls and identified the differentially expressed protein by using mass spectrometry techniques, including Mardy-Toftoff and LC-MS-MS. Finally, the interesting identified proteins were validated from techniques such as Western blots. So, if you remember a few years ago, the severe acute respiratory syndrome occurred in 2002-2003 and thousands of deaths were reported in several countries around the globe. The genome sequence of SARS virus was already known and the structure of main protease and receptors were also known. However, the pathogenesis of SARS was not very clearly understood. Since serum or plasma, they provide a very valuable sample to identify the targets for diagnostic, prognostic and therapeutics. So, authors used plasma sample for the analysis of proteomic alteration in the SARS patients. For the plasma proteomic analysis, authors explored the possible pathogenetic mechanism of progression of SARS by analyzing plasma proteins of 22 different plasma samples, which were obtained from the 4 SARS patient and 6 healthy controls. Authors analyzed plasma proteome by using two-dimensional electrophoresis on 4 to 7 pH immobilized pH gradient strips, and they stained the gels with a sensitivity stain Cypro ruby. So, the slide you can see the steps involved in the traditional two-dimensional electrophoresis followed by the mass spectrometry. Both the controls and the treatments, the healthy individuals as well as patients suffering from SARS, the plasma was separated and then proteins were resolved on the 2D gels. Differentially expressed proteins were further subjected to the in-gel tryptase digestion followed by the mass spectrometry. So, the comparative proteomic analysis of these 2D gels revealed that 38 protein spots were differentially expressed, more than two-fold change. And out of these spots, there were 30 proteins which were upregulated, 35 were upregulated and 3 were downregulated. Now, these interesting 38 differentially and significantly expressed proteins were excised from the gels and subjected to the multi-TOF mass spectrometry for analysis. Authors also employed liquid chromatography tandem MSMS system for analysis of these spots, and then the data was analyzed by using mascot search engine. Now, after the identity of these proteins were established, the 7 proteins which were not detectable in the healthy controls and only observed in the SARS patient, the identity of those included glutathione peroxidase, PRX2, retinol binding protein, vitamin D binding protein and serum amyloid A proteins. They also found that there are 8 proteins which were overexpressed, and those included pigment epithelium derived factor, 2-HS glycoprotein, complement factor H related protein and leucine rich 2 glycoprotein. For complete list of the proteins identified, you can refer to this manuscript. These are some of the interesting proteins which authors identified. After the identity of these proteins were established, then Western blot analysis was used to validate few targets. Among the identified proteins, author found that peroxyredoxin 2 was very interesting. So, they observed that an intracellular protein PRX2 exclusively found in the plasma of SARS patients, but that was absent in the healthy individuals. These results demonstrated that PRX2 can be present in the monomeric or dimeric form. So, they performed the Western blot in both reducing and the non-reducing conditions, as you can see in the slide, and then they loaded different samples including recombinant peroxyredoxin, normal or healthy individual, patient sample 1 and patient sample 2. So, the Western blot result shown that the PRX2 is present in the monomeric form at the 22 kilodalton, as you can see the molecular weight mentioned in the slide, in the reducing condition and in the dimeric form at 44 kilodalton under the non-reducing conditions. These results demonstrated that 4 out of 20 SARS probable cases and 4 of the 20 SARS suspected cases showed higher upregulation of the plasma peroxyredoxin 2. So, among various proteins which were interesting identified from this study, peroxyredoxin 2 was appeared quite interesting, and also because it was validated by the independent techniques. So, the level of plasma peroxyredoxin 2 in patients with SARS was significantly high. Proteomic analysis and Western blot validation suggested that peroxyredoxin 2 may be used as one of the SARS disease associated biomarker. They also found that several acute phase proteins or APPs, those were differentially expressed. The active innate immune responses and oxidation associated injuries may play a major role in the SARS pathogenesis. The acute phase proteins found in the SARS patients suggested that the health status of liver in these patients may be affected their defense against the SARS virus infection. So, some of these information authors are able to obtain because of the proteomic analysis of these plasma samples obtained from SARS patients. Now, in the previous lectures, when we talked about obtaining a good 2D gel image, then you may realize or feel that at the end of that experiment, all the 2D gel image will look very good. So, I am showing you one 2D gel which is appearing very good, and this is something when everything goes very well. So, if your protein separation and staining everything is fine, then at the end you should be able to see very good protein separation on the gel. This is a representative 2D gel image shown with the human serum sample. Now, I am showing you another good gel obtained from bacterial sample, the B subtilis, but often this is not the case. Many times there are different type of issues which could give rise to very bad images. These issues could be because of the sample preparation method, because of reagents involved, because of isoelectric focusing, different type of parameters involved, and finally, different type of staining methods being used. So, let us talk about some of these issues step by step. So, there are various challenges which are associated with two-dimensional electrophoresis. Mainly, the gel artifacts are major limitations. How to compare two gels, how to minimize the variations. Often, during the electrophoretic run, that there is lot of run to run variation. Then, you have different type of technical and biological variations. The image analysis, that itself is a very challenging process. How to define the spot boundaries, how to extract the intensity information from those spots. Often there is a user bias in doing this type of data analysis. So, there are major sources of variations in two-dimensional electrophoresis. I am showing you few problematic gels, how even the small mistakes or some issues inherent to your biological samples can give rise to very bad 2D gels. For example, I am showing you few sample preparation issues. In this gel, as you can see, the TC acetone precipitation method was followed, but there was not good washing performed. So, TCA still remained in the pellet and one can obtain the pattern as shown in this gel. Now, if you are performing the plant protein extraction, but the solubilization is not sufficient, without precipitation, one can see these type of streaking and uneven gel pattern. Now, if you have samples which are having proteins in the abundance, such as crude serum sample. So, these samples will show you different interference in the focusing step because of large salt and other interfering components present, and then you can see these type of bad gels because of the either salt or different type of abundant proteins present in the mixture. Now, the sample variation is one problem. Often the chemical impurities or the chemical ingredients can also give rise to different type of bad gels, such as the chemical impurities shown in this gel, where the urea is very impure and one can see the carbamylation trail as shown in this gel. If the timid is very old, often the timid is very small amount of timid is used in making the second dimension gel SDS page. So, people often use that bottle for very long time, and if timid is very old, you can see some bad pattern on the gel, something similar to as shown in this image. Now, if your chemical such as tris is not very good quality, it is impure, again that will show some artifacts in the gel. Streaking, that is very oftenly seen because of different type of problems which could be associated, whether it is coming from the sample preparation or because of different type of interfering components. Often you have nucleic acid or carbohydrates is still present in the mixture. The extraction method was not sufficient to eliminate all of those impurities. Many times salts and different other interfering components are also present, which interfere in the IF process, and one can see the streaking pattern after looking at the staining of the gels. So, as shown in this gel, the presence of salt can be interfering and may result into vertical streaking as shown in this gel. If you are using narrow pH gradient strip such as 4 to 7 pH strip, then the vertical streaking can appear, because all the proteins beyond 7 pi will stack together in that region and one can see the vertical streaking. Now, abundant proteins, as we briefly talked last time, sometimes crude samples do contain highly abundant proteins and salts. So, abundant proteins are one of the major interfering components which can be easily seen on the gel. If you have serum sample, there are many interfering components present in the serum, including some abundant proteins such as serum albumin. Now, in the plants, one can expect ribosco as one of the very large protein present in the plant leaves. So, these abundant proteins, they mask many smaller protein and create problem in doing the two dimensional electrophoresis, because of IEF process, as well as when you stain on the gel, you can see different type of streaking, as well as different, the whole region is masked because of the abundance of that protein. So, in this gel, it is shown that the human serum protein contains high abundant proteins, which is visible in this area. Now, there are various ways one can overcome these limitations, and as in the previous class of the protein extraction and sample preparation, we have talked about how to overcome these limitations, how to remove the highly abundant proteins. So, please refer to that lecture and one can overcome this limitation. Now, after protein extraction and IEF process is done, one needs to equilibrate this strip. Equilibration process itself can create some problems. For example, if you have performed only first equilibration with DTT and forgot to do the second dimension, the second equilibration prior to doing the second dimension separation, then you can see some pattern similar to as shown here, that only equilibration with the DTT. Many times people think that by increasing the time of doing equilibration, probably the gel quality may appear better, but that is not the case. In this gel, it is shown that by increasing the equilibration time to 20 minutes of DTT and 30 minutes of iodoostamide, the gel pattern looks very problematic, and that is because of the equilibration excess. Now, IF process is very crucial. Many times during the IF run itself, one can monitor the problems if you have a software which is showing you how the run is progressing. Often if your voltage setting is not correct, it is insufficient for the complete focusing, it may result into the under focusing, as shown in this image here for the under focusing. Now, if you have run too much voltage and the overall volt hour is very high, then it may result into over focusing. So, optimizing a focusing protocol for the appropriate duration and overall volt hour is very important. So, there are different type of sorts of variations can be introduced from the biological as well as technical means. There are different biological variations on which there is no control. If you are doing some clinical studies, then even if you have done proper age and gender match, still there will be some variations due to each individual's uniqueness. So, those biological variations are very tough to eliminate, but then there are a lot of technical variations which can be improved during the sample preparation, during the IEF settings, during equilibration steps. So, all of those steps may result into large variations. So, in two dimensional electrophoresis, when you are separating your control and treatment gels on two different gels and you have so many variations coming from both biological and technical variants, then overall the analysis and the confidence in the data becomes very questionable. To overcome those limitations, the new approach, the advanced electrophoresis method, two dimensional difference gel electrophoresis or 2D dyes have emerged.